So today I'm going to be going over the notes on biochemistry. So living organisms are constantly carrying out complex chemical reactions. They are made up of complex chemical compounds. In order to fully understand living things, we need to understand their chemical compositions and their chemical reactions that occur within them. Atoms are the basic unit of matter. They are made up of protons, neutrons, and electrons. A molecular bond holds two atoms together. Energy is needed to make a bond, and energy is released when one is broken. Sharing of electrons is a covalent bond. A molecule is a unit of two or more atoms. Some molecules share electrons unequally. These molecules have a slightly positive side and a slightly negative side. Water being an example of that. And polar molecules dissolve well with other polar molecules and not nonpolar molecules. An organic molecule contains both carbon and hydrogen atoms. An inorganic molecule does not include both carbon and hydrogen. Molecular formula shows the types and numbers of atoms in a molecule. A structural formula is a diagram that shows how atoms are arranged in a molecule. A chemical reaction involves changes in the chemical bonds that join atoms and compounds. A molecular equation shows how the molecules are involved in the chemical reaction and what molecules formed out of the reaction. Carbon, hydrogen, oxygen, and nitrogen are the four main elements that make up over 99% of your body. Carbohydrates are your primary energy source, and some have structural functions as well. Carbohydrates are composed of carbon, hydrogen, and oxygen. There are three types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are the monomers of carbs. They're simple sugars. The molecular formula of monosaccharides is C6H12 and O6. Some types are glucose and fructose. Disaccharides are sucrose, or table sugar, lactose, maltose, and maltose. Polysaccharides are formed by joining many glucose molecules. Starch is a storage form of glucose in plants. Glycerin is a storage form of glucose in animals. Cellulose is found in plant cells and is indigestible by animals. And chitin is found in the exoskeleton of insects. The function of lipids are stored energy, insulation, protection, and cell membranes. They are composed of carbon, hydrogen, and oxygen. Their building blocks are glycerin and 3 fatty acids. Fats are formed mostly by animals. They are solid at room temperature. They lead to cholesterol formation and contain saltwater acids. Some examples are lard, slobber, and butter. Oils are mostly formed by plants. They are liquids at room temperature, and they contain unsaturated fatty acids. Some examples are sunflower oil or corn oil. Some other lipids are cholesterol, steroids, wax, and phospholipids. Proteins are structural molecules found in cell membranes, hair and fingernails, connective tissue, ligaments, bones, and tendons. Enzymes aid chemical reactions. Proteins are composed of carbon, hydrogen, and nitrogen. The building blocks of proteins are amino acids. Protein function depends on their shape. If the shape changes, the protein stops working. High temperatures and changes in pH level can change the shape of a protein. Enzymes are organic catalysts. They are proteins that aid in lowering activation energy and or changing the rate of reaction. Activation energy is the amount of energy needed to start a chemical reaction. It is usually in the form of heat. Enzymes are large globular proteins. The shape of enzymes' active sites closely corresponds with the shape of the substrate. If the shapes are not similar, the enzyme cannot connect to the substrate. Enzymes take on the name of the substrate. You can get the substrate name and replace the ending with "-os". For example, one example of the lifetime of an enzyme is a lactase enzyme. It's generated by a cell when the molecule lactose is present. Lactase and lactose join together temporarily for the lactase active site, forming an enzyme substrate complex. Lactase causes the lactose molecule to break down into its component building blocks. So it breaks down. Once lactase has been broken down into lactose, that releases products and is now free to break down another lactose molecule. If the enzyme shape does not change, they can do the same thing over and over and over again. Each enzyme has a temperature that it functions best at. The higher the temperature, higher temperatures change the shape of the enzyme, which destroys it, or deactivates it, I guess you could say. Lower temperatures just slow down the enzyme and doesn't deactivate it. Humans and animals have enzymes that work best at 38 degrees. Each enzyme also has an optimum pH level it functions best at. Going above or below this pH will change the shape of the enzyme. Enzymes in the stomach like a pH of 2, and enzymes in the small intestine have a pH of 8. Being able to control enzyme activity is critical for maintaining homeostasis. Having too little or too much of a molecule because of an uncontrollable enzyme can lead to illness or death. pH is a measure of acidity or basicity of a solution. It is measured by testing for the number of H+ ions in a solution. More H+ ions leads to acid and less is base. So I hope that helped.